A cross-section of a testis under the microscope shows numerous tubules. Here we see the cells lining one of these tubules. Each lining cell divides, redivides, and changes finally into fully mature sperms. From the testis proper, the sperms pass into a much coiled duct, the epididymis, where they are stored for a time. At the time of mating, the sperms pass up through the sperm ducts, where they are mixed with fluids from the seminal vesicles and prostate gland. These fluids activate the sperms. The fluids also serve as a vehicle for their transportation to the female genital tract. The female reproductive system consists of 2 ovaries, 2 oviducts, 2 uterine horns, and the vagina. Here we see these same organs dissected out. In man, the uterine horns are fused into a single uterus. We shall now take an ovary, prepare a cross-section, and place it under the microscope for observation. Note the large nests of cells, each with a potential egg in its center. Here we see one of these nests growing. Note the egg. When the egg is fully mature, it breaks through the wall of the ovary. In the pig, eggs leave the ovaries every 21 days. Now let us assume that mating has just occurred and that sperms have been deposited in the vagina. Due to the whipping action of their tails and other factors, the sperms will travel up through the uterine horns and finally reach the upper ends of the oviducts. If sperms are there when an egg leaves the ovary, then fertilization may take place. This process of fertilization is best shown by animated drawings. Here is an egg with its nucleus being approached by several sperms. Many sperms may reach the egg, but only one actually enters it. The nucleus of the successful sperm travels toward the egg nucleus and fuses with it. The fertilized egg almost immediately starts to divide. These early divisions we shall show in an amphibian. The picture shows these activities greatly speeded up. Later divisions result in the formation of a ball of cells called the blastula. This stage is followed by a period of development which consists chiefly of cell movements that give rise to distinct cell layers. This stage is called the gastrula. Here we see a cross-section of the gastrula showing its cell layers. In the pig, these early developmental changes occur as the embryo moves down the oviduct. It takes the pig embryo about 6 days to reach the uterus. Here it becomes implanted in the uterine wall and undergoes further development. One of the first sets of organs to appear is the nervous system. We shall show its development in an amphibian. It is first apparent as a thickened plate. Next, 2 longitudinal folds appear. These eventually fuse to form a tube along the midline. At the top of the picture, we see the tube enlarging to form the brain, while the lower part develops into the spinal cord. A pig embryo at 14 days shows similar neural folds. The paired blocks along the sides of the neural folds give rise to muscles and other tissues. The circulatory system also appears early in the development of the embryo. By the 16th day, as shown in this view, the heart is already quite large. Here we see the heart beating in a chick embryo incubated 24 hours. At the same time that the circulatory system is developing, certain important membranes begin to form. Here we see the top view of an early pig embryo removed from the uterus. Certain important membranes have started to develop. Here we see the folds of these membranes growing up around the embryo from all sides. They finally meet above the embryo, forming an outer membrane and an inner membrane, the amnion. Below the embryo is the yolk sac, an ancestral remnant. Note the smaller sac, the allantois. It grows and carries embryonic blood vessels to the uterine wall. The region where the embryonic blood vessels come in close contact with the uterine blood vessels is called the placenta. This close relationship of the embryonic membranes and the uterine wall permits a ready passage of food and oxygen by diffusion from the mother's blood to the embryo's blood, and wastes from the embryos to the mother's blood. It must be remembered that the mother's blood never flows through the embryo. Let us look at this model of a pig embryo after 3 weeks of development. It is then about 3/10 of an inch long. Up to a certain stage, the embryos of all mammals are alike, as we can see by comparing this pig embryo at the left to a human embryo in its 5th week of development at the right. Let us follow the development of the external features in both forms. Note the gradual appearance of differences, such as the facial characteristics, limb features. Also, note the difference in the rate of development. It is an observed fact that the closer two species are related, the longer they parallel one another in development. On cutting open the pig uterus, we see the embryonic membranes which surround the embryo. The inner one is the amnion, which surrounds the embryo with a watery medium. On cutting these membranes, we see the amniotic fluid flow out. Note the umbilical cord, which carries the blood vessels of the embryo to and from the placental area. After 4 months of intrauterine development, the pig embryo is ready to be born. The uterine walls contract rhythmically, thereby moving the embryo until the embryonic membranes break. Finally, the embryo is expelled from the uterus. Thus we see completed the coming into being of a new individual as it occurs in one of our domesticated mammals. In the pig, as in all mammals, the mother continues for a time to supply nourishment to her offspring.